In the 1940s, we knew very little about how traits were passed down. Imagine a vast library of knowledge, but the language was utterly alien. This was the challenge with heredity. We knew genes existed like whispers of a secret code, but their true nature remained shrouded in mystery. What were these mysterious units of inheritance made of? How did they hold the instructions for life? The answer, it turned out, lay deep within the heart of the cell in a molecule called DNA. For a long time, proteins were considered the most likely candidates for carrying genetic information. They were complex, diverse, and seemed to hold the key to life's complexity. However, a series of groundbreaking experiments would soon challenge this assumption. These experiments, carried out by scientists with an insatiable curiosity, would forever change our understanding of life itself. One of the most pivotal moments in this scientific revolution was the discovery made by Oswald Avery and his colleagues. In 1944, they performed a series of elegant experiments using pneumococcal bacteria. They discovered that DNA, not protein, was the transforming principle that could change the characteristics of these bacteria. This was a revolutionary finding. It suggested that DNA, a molecule once thought to be too simple, held the blueprint of life. Then came the Hershey-Chase experiments in 1952, which provided further confirmation. Using bacteriophages, viruses that infect bacteria, Alfred Hershey and Martha Chase showed that DNA was the genetic material injected into bacteria to produce more viruses. These viruses were essentially hijacking the bacterial cells, using the cell's own machinery to replicate. And the instructions for this hostile takeover, they were encoded in the DNA. With DNA identified as the carrier of genetic information, the race was on to decipher its structure. In 1953, James Watson and Francis Crick achieved a monumental breakthrough. They proposed the now famous double helix model of DNA, a structure as elegant as it was profound. Imagine a twisted ladder. The sides of this ladder are formed by alternating sugar and phosphate molecules. The rungs, the connecting pieces, are made up of pairs of nitrogenous bases. There are 4 types of these bases: adenine, thymine, guanine, and cytosine. These bases pair up in a very specific way. Adenine always pairs with thymine, and guanine always pairs with cytosine. This pairing, based on hydrogen bonds, is the secret to DNA's ability to replicate and pass on genetic information. Each strand serves as a template for a new strand, ensuring that the genetic code is faithfully copied. But the story of DNA's structure doesn't end with the double helix. Imagine trying to fit a very long thread into a tiny bead. You'd need to coil it up very tightly. That's essentially what happens with DNA inside a cell. The double helix itself is further coiled and supercoiled to fit within the confines of the cell nucleus. This supercoiling is essential for DNA packaging and for controlling which genes are active. Of course, this intricate coiling and uncoiling requires special molecular machinery. Enter the topoisomerases, enzymes that manage the topological state of DNA. They act like tiny molecular scissors and sealers, cutting, passing, and resealing DNA strands to relieve tension and allow for replication and transcription. These enzymes play a crucial role in maintaining the integrity of the genetic code. The discovery of DNA's structure and the mechanisms that govern its replication and organization marked a turning point in biology. It paved the way for the field of molecular biology and revolutionized our understanding of life itself. From genetic engineering to gene therapy, the applications of this knowledge continue to shape our world today.